Good afternoon, everyone. Let me introduce myself. This is Madan Ambavaram. I'm a senior application scientist at Covaris. In my presentation, I would like to discuss about RNA sharing applications using Covaris adaptive focused acoustic technology. So, in brief, what are the current challenges that we are facing in RNA fragmentation, and what are the existing methods out in the market, and using a couple of customer case studies, and what we Covaris offering for the RNA fragmentation or RNA sequencing applications in general. So, speaking about challenges and solutions for RNA sequencing workflows, as Eugenio already elucidated about the power of AFA in germline and somatic solutions, so I wanted to reiterate here that Covaris has been extensively used for DNA-based NGS libraries, and it has been noted as a gold standard for DNA fragmentation. So Covaris AFA has been used for many other applications. One such application is RNA fragmentation. Here I would like to discuss more about the how Covaris has been used for RNA fragmentation application. One of the challenges what we have seen in the RNA-based NGS workflows is the finding the right fragmentation. Using the non-AFA based fragmentation solution, it is really challenging to find a right fragment sizes for their specific needs of the applications. And also, when we are doing the non-AFA based fragmentation, there is a chance and losing the more RNA. So how one can protect the integrity by without losing the much of the sample. So that is also one of the challenging. The Covaris based AFA solution has a like how we can optimize the fine fragmentation and tunable solution. Speaking about the sample variability, as we know that we are speaking about the clinical samples and the environmental samples, sample to sample variability is a kind of it's a common. So how we are going to address those challenges considering the sample to sample variability? Covaris AFA is sample agnostic, and I will be discussing more in the subsequent slide. And also the compatibility of the various workflows. So ribosomal rRNA depletion is one of the critical steps in the RNA sequencing workflows. So by how we can consider using that AFA and the RNA based workflows. And also speaking about the implementation. So for example, using the non-AFA based application, it takes more long optimization time, which is more challenging considering the sample quality in the clinical samples, and also lack of streamlined workflow. Since we are talking about the high throughput solutions for larger scale and optimizing sample to sample, it is really tedious and taking a more time. Translating all these solutions, which significantly saves the cost. That is what we offering at the Covaris. So, having said that, what are the best RNA fragmentation solutions that we are offering? As Eugenio has already mentioned, the three pillars of our Covaris. One is the workflow ease of use. Speaking about the scalability, so Covaris has a solution from one sample to up to 384 processing samples. And also speaking about the efficiency with the minimal optimization or no optimization across various sample types, and also fast turnaround time by minimizing the sample cleanup steps and other various heat steps and PCR workflows. Speaking about performance and economics, AFA is a game changer for many reasons. To name it, for a better sequencing quality, which we have seen from the customer studies, more than 99.8% of the mapping rates, and also reproducible sequencing data, which helps to quantify the rare events and the fusion event. And these are all translating into the cost, which is significantly by improving the depth of the sequencing and for lower sequencing failure rates. So these are all helps to improve the significant cost savings. Here I would like to take a minute to explain what are the additional challenges compared to the DNA sequencing. Having a tight fragmentation is a critical step, either DNA or RNA fragmentation. Besides that, requirement to convert the cDNA and also ribosomal RNA depletion. There are various protocols that are existing out in the market, and how these are going to having the challenges and all. There is only, speaking about the RNA fragmentation, currently almost all kits that use the cation based fragmentation, which is in a combination of magnesium and heat. As we all know that this is very sensitive to initial concentration, how much we are using. Speaking about the sample to sample variability and considering the right concentration, as you can see here, one of the this graphic explains, which is came from the recent review paper. Finding the right concentration, which is challenging, sometimes it takes forever. And also using the too much or too little magnesium accelerate the RNA cleavage, which has a significant downstream effect in terms of the library, making a library and also the sequencing effects. Here we are offering Covaris. AFA based fragmentation is an alternative to the existing chemical fragmentation method by ensuring the high quality libraries. Here is an example, the representative example. Like I have mentioned, AFA is agnostic to the sample type. The RNA comes from either cell culture or FAPE tissue or mRNA or double-stranded RNA. Here is the top gel electropherograms. You can see that, okay, the represent after AFA treatment, how tight fragmentation we can obtain, irrespective of whether it is a degraded RNA sample or the intact RNA sample like mRNA and double-stranded RNA. The key takeaways here is that here is the sample is agnostic to the sample type, and also the concentration independent. That is the beauty of for the NGS workflows, which is concentration independent, and highly tunable fragmentation, and also scalable for efficient workflows from sample 1 to 384 samples in one run. Here is one of the application and the case study. Our customer and collaborator, Massachusetts Institute of Technology Biomed Center, they have used Covaris for their tumor transcriptome studies. Here, as you can see here in the top left gel picture, which is represented about 69 FAP RNA sample, which is highly degraded. As you can see, the sample quality is highly variable. Using these kind of samples as minimum as 2 nanograms, they have obtained the Illumina libraries. Here, this gel picture which speaks about how homogeneous the libraries are, which were shared using the Covaris RNA sequencing. This is clearly demonstrate that AFA-based fragmentation is a non-destructive and also insensitive to the input amount by ensuring the high quality of the libraries. This study has been used for developmental deconvolution for the identifying the cancer origins, that has been published in the peer-reviewed article, The Cancer Discovery. Here is another application where they have used Covaris for how we can say that they are finding the uniform RNA shearing, which leads to the improved transcriptome assembly. They have done this study very diligently using the both cation fragmentation as well as the Covaris based fragmentation, using the two sample types. One is the intact RNA and also degraded RNA sample. The Covaris based shearing, here you can see that the sesame plots in the blue here, which clearly demonstrate that there is increased expression of certain gene cluster, which is a critical regions of the genome. And also they have observed the log ratio by increased transcript expression and also consistent mean expression, particularly for the various GC regions, and also identified the UTRs, the 5' and 3' UTR regions. This data clearly demonstrate that Covaris shearing is a consistent and uniform sequencing result even in the degraded samples. Here I wanted to bring your attention that on November 14th, there is a webinar. Stuart Levin, who is a director for MIT Biomicro Center, is going to speak how Covaris RNA shearing enabled in their workflows. this is just a teaser, and I highly recommend and request you to register for this webinar, and we are happy to help. Please stop by at our booth, Covaris booth. Here is another application as we speak about in the mRNA vaccine world. So the Covaris sharing is also used for the mRNA based library prep. The customer at Greenlight Biosciences, they have used Covaris sharing for they have observed the tunable fragmentation, which is pivotal for successful library. And how this is directly proportionate to the finding the high quality libraries. They have obtained a high Q score, which speaks about the direct performance and less sequencing errors, and also reproducible sequencing data, which is superior coverage and uniformity. And also they have observed, when you speaking about the sequencing matrix, they have find the context where it is a critical region and the better sequencing regions. Using that, they are using the various settings, and they have find the right fragmentation, which help to identify better sequencing and better coverage in the critical regions. And the customer from Greenlight Biosciences, they have also used Covaris for their double-stranded RNA fragmentation. Their objective is for the Illumina sequencing to find the sense and antisense, the preferential strand sequencing data. Here you can see the plot. They have the alternative way of fragmentation is the DNA. They have to find the different concentration, which is really challenging, as this plot clearly says that. Using the Covaris fragmentation, they could find the right fragmentation size for the large scale of the samples, where using the DMSO based and heat based, they have challenges where at one point they even found that plateau, they couldn't able to get the right fragmentation which is required for the library prep they are using. And also speaking about the coverage plots, they have obtained more than 99% of the sequencing read, the sense and antisense, which is their objective to find, and how we can switch the finding the right sequencing data for the strand specific sequencing. The Covaris offering for RNA sequencing application, there are two instruments and high throughput capable. You can use from the sample 8 to 94. We have a 96 AFA TPX plated option, and low throughput, mid throughput, and high throughput solutions. This can be used. The key takeaways here is that this is the single fragmentation workflow for various sample type, which required minimal or no optimization. And also speaking about the cost savings, this is highly suitable for high throughput RNA library prep and also for the miniaturization workflows, which is highly compatible for various ribosomal RNA depletion workflow, which speaks about all-in-one solution plugging into the broad spectrum of RNA sequencing workflows. Here are, like as we discussed, various sample types, and also has an option like the sample volume. We have optimized protocols right from 10 microliter to 20 microliter. So here, like the trajectory of using the Covaris for RNA fragmentation solution, here are some of the key customers where they have been using Covaris for their RNA fragmentation and RNA sequencing workflows, like besides MIT, Sanofi, Roche, and Lakmine Technologies, they have been using the Covaris for their RNA sequencing workflows. This has been published in the technical notes and also peer-reviewed journals where they have used the Covaris for their RNA sequencing applications. With this, I would like to thank all for your attention, and I open a session for Q&A. We are happy to take questions, and thank you all.